Hello, dear professor. Today we are going to talk about the regenerative potential of nanomaterials in hard tissues. It's me, Amal Mustafa Slim, Assistant Lecturer of Oral Biology, Faculty of Dentistry, Cairo University. The ability of the human body tissues to repair itself and heal back to its original condition after injury or infection or any other type of insult is a natural process, though the healing is actually a limited process, even though it's a natural but yet limited. So when the injury or insult is causing tissue destruction beyond the healing capacity, here comes the role of regenerative dentistry or regenerative medicine, and this is considered one of the challenges for tissue engineering and regenerative medicine, or we can call it as TERM. And this is importantly known as engineering, or comprised of three important things: the materials and the tissue itself, or tissue architecture, along with the engineering cells. So we can conclude that the TERM is consisting of three important items that are the scaffolds and growth factors and the cells. This is the tissue engineering construct. So the stem cells, which is the base or the seed for this triad, and the scaffold that provides support, and growth factors are actually the driver that can drive the cells to differentiate into any type of cell according to their guidance. So, what this is related to our topic for today, the nano materials used in tissue engineering. Well, we got nanoparticles emerging in this field of TERM is actually having a great effort today with the nanoparticles in special. They can be used for cancer treatment, for drug delivery system, or even can be used as delivering growth factors to tissues, or even genetic material delivery, as we are going to see along with the path of this or sequence of slides. Let's first talk about the regenerative potential of nanomaterials in bone tissue. Well, sometimes there are defects or bone damage occurs with defects, whether from infections or birth defects or even from trauma, that are beyond the healing capacity of the body. And here comes the way where we need the regenerative potential to be emerging in this. Actually, the conventional surgical methods like autografts, allografts, or even synthetic biomaterials are not without shortcomings. And that's why we needed the nanoparticles or nanomaterials to emerge in the field of TERM or tissue engineering. Now, let's move to what is the importance of nanotechnology in bone tissue engineering. What is the effects or the perfectly provided effects after the emergence of nanotechnology? The first item for today is to be suitable mechanical properties. Not just mechanical, actually, chemical and even surface properties can be affected greatly with the emergence of nanotechnology, and that subsequently giving good tissue support. Also, they are improving the osteoinductive and the osteoconductive potential. Also, they are providing non-toxicity and enhancing the biocompatibility, biodegradability, and allow high absorption properties of the materials used. Also, the nanoparticles or nanoparticles based dentistry nowadays emerging in the imaging technologies, and this is for tracking tissues or tracking the cells used in treatments, and this is also very important. Also, they are providing release of drugs and certain growth factors. So let's see how to apply this in tissue engineering. What is the applications of nano materials in general in bone tissue engineering. In this diagram, we can see that the nanotechnology is emerging in being as carriers or scaffold or even in imaging. For being a scaffold, the nanocomposite scaffolds are providing the support or the structural support for the cells. The nanoscale features have been shown to have regulatory effects over the multiple aspects of the osteoblast, especially osteoblast and bone-derived stem cells behavior, including adhesion, migration, proliferation, cell signaling, expression of the genetic matter, and even the fate of the cells. As we can see here in this diagram, we can see that the different array or geometry of the scaffold can affect the fate of the cells. The cells can actually differentiate into any type, not just by the presence of certain types of growth factors, but also one of the most important items is the geometry of the scaffold. So here comes the first important rule for the scaffolds to affect the fate. We can use the nanomaterials in case of drug delivery or gene delivery systems. We can use it as drug delivery to deliver certain types of drugs to the tissues in cases of treatment of bone, especially in bone defects. Not just bone defects in general, but also they can be used in case of cancer treatments, as we see here. Also, in a recent paper or a recent study, bone cement modified with nanosilver or even nanocopper can show higher bactericidal effect in antibiofilm formation, and this is compared with the conventional antibiotic-loaded bone cement gives better results. Also, the cement containing nanosilver particles doesn't influence the erythrocyte hemolysis or of the blood platelet functioning, and the viability or morphology or even the adhesion of the dental pulp stem cells, and this is really affecting the outcome or feedback from the tissues. We can conclude that using it or the nanoparticles can affect the healing power, especially in case of dental pulp regeneration, as we are going to see later. Also, we can use it for gene delivery. Of course, we know that we can use the gene delivery system to deliver certain genes, especially in cases of bone destructing diseases like cancers or that destroy the tissues beyond the healing power of it. So we can adjust the DNA or RNA or even any type of genetic matter to affect the tissue. Now let's move to what are the different nanoparticles currently used on bone tissue engineering. We have so many items today or to talk about today. As we can see here, the liposomes are having so many advantages, but the most important one of them is that it's biodegradable and stimuli responsive, can be incorporated by hydrophilic or even hydrophobic drugs for drug delivery system, but they can be rapidly cleared from the tissues or the circulation. Yes, this is the conventional liposomes, and they may be advancing this recently. We also can see the polymeric nanoparticles. The polymeric nanoparticles are like liposomes. They are biocompatible and biodegradable, but they scale up the tissues also. We can see also the dendrimers that are biocompatible with large number of surface functional groups, but they are unfortunately having one of the most importantly items we look for today, especially in using any type of drugs or materials in human body. That is toxicity. So it's size dependent toxicity. Yes, it can be controlled, but with poor drug retention in it, and this affects the use of the dendrimers nowadays. Also, we have other types of particles today, like as we can see in this diagram, we can see the silica nanoparticles. They are of uniform form with biocompatibility, and the chemical stability is perfect, but they are of unfortunately variable toxicity, and this makes its use a little bit affected by this item. Also, we can see the gold nanoparticles that are biocompatible with reduced cytotoxicity and easy functionalization of it. We can also see the magnetic nanoparticles here. They are long lasting and non invasive tracking with super paramagnetic properties, but yet they are causing a little toxicity or a little more toxicity than the previous items, and their coding is a matter of importance and is required as a must. Also, we can see other particles like the ceramic nanoparticles, and they are bioactive, bioresorbable, biocompatible, and they can improve the mechanical properties of the scaffolds, but they are having complications during the synthesis that may affect the particles itself. Also, the nanotubes, the carbon nanotubes, which is having two sides for functionalization, and they can also improve the scaffold material or structure, but they are cytotoxic to the tissues as well. Also, we can see the quantum dots. They are having a photostability with multicolor imaging and bright emission, but they are having toxicity issues, and their coding is a must. We just said that we can use the nanotechnology in scaffold material and use it as a carrier for either drug delivery or gene delivery system, but also we can use it in imaging technologies. Because recently the exogenous therapy aims to replace or repair the diseased tissues, and they give promises to regenerative medicine. So, to track the cells used in cell therapy through their location, distribution, long-term persistence of these cells, this can be evaluated by the imaging using the nanomaterials. Now, let's see the next item for today, which is what is the importance of nanobiomaterials in dental implants? Along with the evolution of nanodentistry, implant dentistry modifications with the nanomaterials actually followed the rule of mimicking the nature. It was proposed that the implant surface may imitate the bone-like structure properties and the properties of the nanoscale level, as bone is nothing more like a biological nanocomposite. So, as we can see here in this diagram, we have two important items. The primary interlock that corresponds to the mechanical anchorage and mostly the macro design of the implant, such as screw shape or threads, and also the bone remodeling or at the implant surface or implant interface, and the creation of biological bond. As we can see here in this diagram, we can see to the right where a specimen for a 2-month complex of the implant and the bone. But the newly formed bone, yes, present, and the connective tissue and the bone marrow detected. But actually, there is still no bone formed or observed at the space between the implant and the bone. Therefore, osseointegration is not achieved. While for the next specimen, where we can see a specimen of 3-month implant surface along with osseointegration is achieved actually. As we can see here that there are implant bone interface. For the last specimen, it's a 6-month complex where showing similar bone formation to that of the 3 months, but actually with increased or clear increase in the bone formation between the implant interface and the bone. Also, we can see here to the left, we can also see how the bone is also formed along with the non-formed or fibrous tissue formed in between the implant bone interface. Now, let's move to the factors that together, when put in considerations, can have an impact on the dental implant. Actually, we have so many items, but we can focus on all the important two that are the surface topography, micro nanotopography, and the chemical composition. Now, what are the surface modifications that can affect the roughness of the implant, like direct surface tailoring, like etching or sandblasting, functioning the surface with like bioglass coating or polymers or coating in general, or even combination of these techniques together to the commercially available implants to achieve the roughness at the micro level. Also, the nanotechnology, which is the most important one for today, because as long as the surface properties have a key role in the long-term stability of the bone tissue, the emergence of nanotechnology in the field actually affected the roughness. So, from a biological point of view, the roughening of the surface of the dental implant is really important because it affects the optimum cellular reactions over the surface that takes place over the surface of the natural tissues, and this affects the proliferation of osteoblasts, and this, of course, would finally affect the osseointegration. So again, the dental implants coated with nanomaterials can affect the proliferation of certain items of cells like osteoblasts and subsequently affects the osseointegration, the whole process that is limited and linked to the nanomaterials is affected greatly here. So now let's move to another item where we need to talk about the recent nanostructured implant coatings used nowadays. We have so many items used for today, like the nanostructured diamond. This coating also represents a very high hardness, improved toughness over common microcrystalline diamond with low friction and good adhesion. And this is a really important item for this one, because the good adhesion of osteoblast to titanium alloys is the biological point of view that makes this item important for today and subsequently affecting the osseointegration. Also, we can see the nanostructured coatings of hydroxyapatite. This type of coating material is applied to the favored mechanical properties, also enriched the surface reactivity, and has shown increment of osteoblast adhesion. We also see the nano-constructed calcium-coated titanium implants with great responsiveness of bones surrounding the implant. Also, the metalloceramic paste nanostructured coatings, like using or coating the titanium implants using zinc, calcium, and phosphate nanocrystals. These coatings are biocompatible as well as can be antibiotic coated and incorporated in the tissues. Also, we got the nanostructured ceramics and the nanophase zinc oxide and titanium oxide coatings. For the nanostructured ceramics, they can enhance the osteoblast adhesion and calcium phosphate mineral adhesion, and this is from a biological point of view can affect the osseointegration greatly. Now, let's move to dentine pulp regeneration. There are so many approaches to engineer the lost dentine and pulp, even if the odontoblast has been lost due to the curious process or even any insult. But recently, we got wide range from the simple nano powders to nano carriers have been introduced to the field of dentine pulp complex regeneration. We got two paths here, as we can see in this diagram. We got cell transplantation along with the cell homing. The stem cells in cell transplantation are isolated and cultivated in vitro, increased in number, and then then inserted in soft scaffolds or even hydrogels with or without the addition of signaling molecules and calcium phosphate nanoparticles, and at the last they are implanted in the receiving site. But for the cell homing, it's not necessarily important to manipulate the cells in vitro, because it's all about reaching the cells in situ, and this happens through stimulation of the residual stem cells by means of molecules by hydrogels in situ. For the cell transplantation, we are able to control the cell number, and the availability of the cell source is in the peripheral defects is okay, or can be helped. And there are utility of subpopulation of stem or progenitor cells, while for the cell homing, nothing of this can be applied actually. But also we have barriers today. For the cell transplantation, we have a problem with the cell isolation from the patient is really a required step. And also we got subsequently could be susceptible to immune rejection. Also contamination could happen. Also, they could induce tumor genesis, and also with along with high cost. While for the cell homing, not that much barriers, but they can show shortage of cells. Recently in endodontics, we got two important concepts that should be taken into considerations today, that are the pulp revascularization, which is the induction of angiogenesis in canals, on root canals, and we got the pulp regeneration. The pulp regeneration, which is a combination of pulp revascularization along with the restoration of the functional odontoplasts or nerve fibers. As we can see here in this diagram, we have three important crucial steps for the pulp regeneration. The first step is that the damaged tissues should be safely disinfected, and all the microorganisms must be eliminated by well-targeted antimicrobials. The second step is to control the inflammation at different levels of the tissues, or we mean that the inflammatory phenotypic cells, destructive exudate, and the damaged extracellular matrix, they all must be gone. And the final step is the regeneration of the lost pulp tissue. And this takes place by developing the type 1 collagen matrix with the fibroblasts, also innervation, vascularization, and odontoplastic layer should be present. For this last step, the endodontic regenerative active biomaterials must promote the colonization and the proliferation of the cells at the peripheral part, just like to mimic the natural appearance of the dental pulp itself. So to reach this all, we need so many items to put in considerations, like the materials. And this is the most important item for today. What are the items to put in considerations using the nanomaterials? The nanomaterials actually provided three important items. They provided being a reservoir for the antibacterial and anti-inflammatory molecules, as we just mentioned before. They can deliver growth factors that are important for the cells to differentiate and proliferate, and also they provide a scaffold in case of dentine pulp complex, as we are going to see later in full details. Number one is the nanoassemblies targeting the first step of the dental pulp regeneration. Nanomaterials for dental pulp regeneration can be used alone or even with growth factors, drugs, or stem cells. But the nanobiomaterials for dental pulp regeneration also allow the concentration of many different functions in a small volume, and they control the cost and delivery of quality targeting molecules. These endodontic nanomaterials can be a reservoir for antibacterial and anti-inflammatory molecules, and they deliver growth factors guiding the migration, proliferation, and differentiation of different pulp cells like fibroblasts and vascular or nervous cells along with the odontoblasts. The second item is the scaffolds. As we can see here in this diagram, we can use it to deliver the material and do just revascularization, or we can add a scaffold material along with the cells to enhance the revascularization along with the scaffold, or even we can use the process in the way of sheets, as we can see to the right of the diagram. Also, incorporation of active biomolecules inside the scaffold allowed orchestration of the tissue regeneration and attraction of the resident cells according to the cell homing process. Stem cells cultured in the scaffold optimized the pulp regeneration, notably the matrix deposition. As we can see here in this diagram, uh, we can see we can see the nanoparticles that or the nanofibers showing a natural appearance or mimicking the natural appearance, allowing for the revascularization along with the blood vessels and nerves, and also the homing of the cells in place, and so finally can reach the target. Now let's move to the nanofibrous and microporous membranes. Different types synthetic and natural polymers are used today to build the nanofibers by electrospinning techniques. The nanofibers of diameters closest to that of the collagen nanofibers, which is about from 50 to 500 nanometers, were prepared. And the electrospins randomized nanofiber network and created micropores that is mimicking the pattern of connective tissue matrix as we can see in the diagram A to the left. The electrospun matrices of PCL or poly-E carbolactin showing favorable results for connective tissue regeneration, and that's why they are going to be used widely later today. The capacity of these matrices to be functionalized showed a lot of success. Now let's move to this item, which is BCL gelatin scaffolds with nanohydroxyapatite. Actually, different nanofibrous microporous scaffolds provided an excellent environment for dentine regeneration recently. The electrospun PCL gelatin scaffolds with nanohydroxyapatite enhanced the differentiation of dental pulp stem cells. So the nanofibrous poly-L-lactic acid or PLLA scaffolds functionalized by polymorphogenic protein 7 and dexamethasone promoted the dentine regeneration, as we can see in this diagram. The mineralization of electrospun PCL scaffolds is particularly attractive for dentine tissue engineering, and this is happening by promoting the growth and odontogenic differentiation of human dental pulp stem cells. The incorporation of mesoporous bioactive glass nanoparticles inside the nanoporous PCL gelatin matrices also enhances this odontogenic differentiation. And as we can see here in this diagram, where the nanofibrous gelatin magnesium scaffolds provided a controlled release of metallic ions, which enhanced the dentine regeneration. Finally, but not final, let's move to enamel regeneration. But before we talk about it, we need to know that it's really difficult to regenerate an acellular, avascular, and non-vital tissue. It's very little research has been published on this tissue engineering of enamel. Therefore, the focus of enamel regeneration research is mainly concerned with the remineralization of the demineralized or defective enamel. The first one is in regeneration about cell-based strategies, where we actually do use synthetic or bioactive nanostructures that are known as or known to be self-assembled in physiologic environments into nanofibers network. This is to mimic the extracellular matrix that surrounding the ameloplasts. The growth factors and cytokines provided insight into molecular mechanisms involved in enamel formation, and this helped in designing synthetic regenerative approaches. The second technique for today is the entire tooth regeneration. Currently, there are two followed techniques or routes for regeneration of entire tooth that are the scaffold based and the stimulation of the embryonic development of natural teeth. As we can see here, We do a selection of the dental cell source, and then we add them to growth factors and culture them in vitro, and then we implant them in place of messed teeth where they can continue growing into natural tooth in the position of the lost tooth. Or we can do that and isolate the cells and do the process as a whole and cultivate the cells from cell lines and continue the same process with the same sequence as well to reach the final result, which is cultivating or forming in a full tooth or entire tooth. But it remains actually to be determined whether such bioengineered teeth can achieve three important items to put in consideration, that are the masticatory function, biomechanical cooperation, and the sensory response of their naturally formed counterparts. And now we have reached the end of this presentation. Thank you so much for listening, my dear professor.